Welcome to everyone who's just logging on today. We're just going to give it about a minute for folks to get connected here. All right, it looks like we've got quite a few people logged on, so we will begin. Hello, everyone, and welcome to today's SSR webinar. If you have any questions about this webinar, feel free to reach out to us directly at SSR headquarters via email at membership@ssr.org. This webinar is scheduled for 60 minutes and it is being recorded. It will be posted on the SSR website shortly thereafter for members to access at their convenience. In just a moment, our moderator will begin with a brief introduction followed by a speaker presentation. Please feel free to submit your questions during the presentation using the Q&A text box in the bottom middle of your screen, and our moderator will read them out to our speakers when the question portion of the webinar begins, and then we will begin with another introduction and our second speaker presentation followed by questions for that. And with that, I will turn things over to our moderator for today.
Thank you, Erica. Hello, everyone. I'm Shuo Xiao from Rutgers University, the moderator for today's webinar. So at first, I would like to represent the SSR Virtual Education Committee to welcome you all to the 2024 Rising Stars in Reproductive Biology webinar series. The virtual event aims to introduce and highlight novel and also cutting-edge research from the new investigators to both regular SSR member and also to a broader reproductive science and medicine community. So today we will have two rising star speakers, including Dr. Haiqi Chen from University of Texas Southwestern Medical Center and Dr. Fei Zhao from University of Wisconsin in Medicine. So let me introduce our first speaker, Dr. Haiqi Chen first. Dr. Chen received his PhD from a joint program between the University of Hong Kong and the Rockefeller University, and completed his postdoc training at the Broad Institute of MIT and Harvard as a Layler Fellow. And Dr. Chen is now an assistant professor at the University of Texas Southwestern Medical Center. And today, he will introduce his research regarding how to use the cutting-edge spatial transcriptomic technologies to study the spermatogonial stem cell lineage. Haiqi, I will turn it over to you.
All right, thank you, Shuo, for the introduction. Let me share my screen now. All right, I would also like to thank the SSR for the opportunity to allow us to present our work. So as Shuo mentioned that, oops, there we go. As Shuo mentioned that our lab focuses on developing and applying cutting-edge genomics tools combined with computational approaches to understand human reproductive health. One of the key questions we're interested in is to understand the molecular mechanism that regulate the proliferation and differentiation of spermatogonia stem cells, or SSC. This audience apparently doesn't need introduction into spermatogenesis or SSCs, but I do want to emphasize that SSCs reside in a tissue environment that really is benefiting from the interactions between the somatic cells and the SSCs themselves. And numerous work previously has proved the way for understanding the SSC niche, and it's important because it allows us to really make strides towards in vitro gametogenesis for fertility preservation and infertility treatment. As I mentioned, previous work has identified a few key ligand receptor interactions that allow the stem cells to proliferate and differentiate in vivo. But those approaches require genetic models as well as cell cultures. And it has been challenging to really canvas the whole landscape of gene expression in the testes, mainly because there's so many genes in the testes. And recent advances in single cell sequencing has allowed us to partially overcome this challenge by looking at cellular heterogeneity in each single cell of different cell types in the testes. But because tissue dissociation, we really lose the spatial context that allows the spermatogenesis to take place. So this this leads to a lot of negative positives, which means that some genes or signaling pathways identified by single-cell sequencing may not necessarily mean that they are important in the context of spermatogenesis or SSC differentiation or proliferation. So in this talk, I'm going to share with you 3 variants on how we leverage spatial transcriptomics to understand the regulation of SSCs in vivo. And the technology that we use to do that is called SlideSeq. It has been commercialized by a company called Curio Bioscience. But to be clear, my work is not supported by the company, but maybe they should. So this technology, the core of the technology is basically this densely packed bead array. Let me change my pointer real quick. So this bead array is composed of thousands of beads, and each bead contains oligos that have unique spatial bead barcode that is unique to the bead, as well as poly T-tails that's used to capture the mRNA from the tissue. So before one can use this array, the array has to go through a spatial indexing step where the bead barcode is rest in situ so that you know exactly where the bead is localized in terms of XY localization, as well as the bead barcode. So for the end user, what you need to do is just to obtain those spatially indexed array and put your tissue on top of the array. And you perform a library preparation that is quite similar to the single-cell library prep. At the end of the day, after sequencing, what you get the data back in the form of 2 matrices. And the first matrix is a gene expression matrix that is quite similar to the single-cell RNA sequencing gene expression matrix, where each row represents a bead. You have your bead barcode, and then each column represents a gene count, and you have the gene count data for the whole transcriptome. And the second matrix is a bead localization matrix that we previously obtained from the spatial indexing step. So basically, those 2 matrices are all you need for downstream analysis. Here I'm showing you an example of a digital reconstruction of the mouse testis cross-section. And each bead is colored by its assigned main testicular cell type. And I hope you can appreciate those circular structures across the digitally reconstructed cross-section. And those circles represent seminiferous tubules. And of course, we can zoom in to look at the spatial distribution of different cell types that was assigned. For example, the spermatogonia, which contain the stem cell population, reside at the periphery of each circle. And whereas the spermatids, which are the haploid cell types, localized near the center of each circle or seminiferous tubules. Again, from the germ cells, you can also look at the distribution of somatic cells, such as the testosterone producing Leydig cells, which form clusters in between space, which is called interstitial space between different seminiferous tubules. And so now with advances of computational approaches, we not only can assign cell types, but also assign cell states within the spatial data. So in this slide, by leveraging public available single cell data, we were able to assign 4 different transcriptional states within the spermatogonia population. And we assigned them SPG 1 to 4, with 1 represents maybe the undifferentiated spermagonia based on the enrichment of gene markers. And SPG 2 to 4 represent differentiating spermatogonia populations. So as I mentioned earlier, one of the key motivations that we want to use spatial transcriptomics to really understand the crosstalk between the somatic cells and SSCs in a native tissue setting. So in this case, we are really interested in looking at the ligand receptor interaction in a spatially resolved manner. So by using an established computational pipeline, we were able to calculate the strength of each ligand receptor interactions among different cell types using the spatial data. And here I'm showing you 2 examples of visualization of those ligand receptor interactions. And so here, there's 2 classic ligand receptor interactions that are known in the testes, the GDNF signaling and FGF-based signaling. You can see that their spatial expression seems to be enriched at the periphery of the tubules, suggesting that it is mostly at the stem cell niche at the basal membrane of the seminiferous tubules, which is consistent with the biology. So one advantage of this approach is that it now allows you to look for ligand receptor interactions whose expression kind of focused only on a specific developmental stages of spermatogenesis. So for example, here I'm showing you on the right 3 examples of those ligand receptors whose expression seems to be only enriched in certain seminiferous tubules, but not the others. And these 3 examples tend to be enriched at stage 4 to 6 seminiferous tubules. Another key advantages of this approach is that it allows you to identify cell type specific ligand receptor interactions. In this case, we are interested in the stem cells. We are looking at the SPG1 population. We are asking what are the differentially expressed ligand receptor interactions coming from different cell types that are surrounding the stem cells. And for example, here, I'm showing you 3 examples of ligand receptor interactions that we found are enriched in the lattice cell stem cell interactions. Again, these ligand receptors seems to be enriched at the basement compartment of the seminiferous tubules. So now, at this point, you may wonder, how do I know this information that we obtained from the spatial data is true or not? And to this end, we proceeded to validate some of the ligand receptor interactions that we found using our spatial data. In this case, we are looking at pleiotrophin or PTN mediated ligand receptor interactions. We found that the PTN seems to be interacting with cytokine receptors in the SSC microenvironment. We first use an IF to look at the co-localization between PTN and cytokine 4, and they seem to co-localize together on the surface of undifferentiated spermagonia. And then we perform a gold standard transplantation assay in collaboration with Nagato lab in McGill University. So in this experiment, what we do is basically we treat the cultured SSC, mouse SSCs with or without PTN for about a month. And then we transplanted the SSCs into recipient mouse testes that do not contain any native stem cells. And then we observe the localization and colonization of those transplanted stem cells. And in this case, what we found was that when you treat the SSCs in culture with 5 nanograms per ml PTN, it seems to be facilitating the colonization of the SSCs in vivo. But if you give the stem cells too much PTN, in this case, 50 nanograms per ml, it seems to actually decrease the colonizing potential of the SSCs. This experiment suggests 2 things. One, PTN does do something in terms of the SSC proliferation or even differentiation. The second, there seems to be a dose-dependent effect on the SSC colonization potential in vivo. This might be because when you add very high dose of PTN, those PTN may be competing with other ligands such as FGF, which is a known binder to cytokine receptors on the surface of SSCs as well. So the last thing I want to demonstrate is, besides physiological conditions, this approach can really be leveraged to understand disease mechanisms. In this case, what we're interested in is to look at diabetes-induced testicular injury. The reason we are interested in diabetes is because it has been known for quite a long time that patients who suffer from diabetes may also have fertility issues, but the underlying mechanism remain largely unexplored. So in this case, we use spatial transcriptomics to profile testes from both the wild type and diabetic testis samples from mouse models. And then hopefully you can already appreciate that there is drastic differences in terms of tubule structures in the digitally reconstructed mouse testes. And we developed a computational pipelines that allow us to really identify ligand receptor interactions whose spatial expression pattern change between diabetic samples and wild type samples. Here I'm showing you 4 examples. On the left, This is the wild type testis data, and the right is the diabetic sample data. And you can see that their spatial distribution changed dramatically. At this point, one may argue that this is obvious because you already showed that in the previous slides, there's a drastic difference in terms of the structural tubule structures. Of course, you're going to see differences in terms of spatial distribution of ligand receptor interactions. But turns out this is not 100% the case because we out of all the ligand receptor interactions that we characterized, there are only a very selected subset of ligand receptors whose spatial expression pattern get changed. In fact, 90% of ligand receptors that we looked at, their spatial expression pattern remain the same. Here are 4 examples. Again, left is wild type, right is the diabetic sample. As you can see that their spatial distribution do not change at all. So this suggests that there's only a selective subset of ligand receptors whose spatial expression pattern were influenced in a diabetic setting. So we further ask the question, what are those genes? What do they do? In this case, we performed a simple GO-term enrichment analysis. And to our surprise, we found that those genes are predominantly enriched in immune cell proliferation or interactions. This seems to suggest that the diabetes really can cause testicular damage through inflammatory responses in the testes. This is a mechanism that needs to be further explored. So to sum up, I've shown you today that we have leveraged spatial transcriptomics to establish this ligand receptor interaction map that allow us to detailing the interaction between SSCs and their surrounding somatic cells. And they also allow us to nominate and further identify novel niche factors that may contribute to the differentiation and proliferation of stem cells. Lastly, they can be used not only for physiological settings, but also for disease settings as well. In the last few minutes, I want to give you a sneak peek of what we are doing now and what we think the field can go from there. I hope that in my previous slides, I've been trying to convince you that spatial transcriptomics is a really powerful tool, allow us to nominate a gene or signaling pathways that may be important in a biological process. We all have our own favorite genes or signaling pathways. So spatial transcriptomics really allows us to, get rid of the bias and look at the data from a more systematic view. But it really depends on downstream functional characterization of the genes or signaling pathways to allow us to really understand what do they really do. So this means that the key bottleneck here is to perform functional assays such as transplantation assays or genetic models or what have you to characterize the genes that are being nominated by the spatial transcriptomics approach. So we got thinking, what if we can make this part of the work high throughput or systemic as well? This brings us to CRISPR screen, a technology that is very powerful to allow systematic gene perturbation and functional characterization. So basically what CRISPR screen does is to introduce a pool of guide RNAs into a pool of cells, and each cell receive a single guide RNA targeting one gene of interest. And in this way, you'll be able to read out individual perturbations using single cell sequencing so that you can match the perturbation with the phenotype at molecular level. But there are caveats associated with CRISPR screen, especially in the context of understanding testicular gene functions, mainly because one, most of the CRISPR screens have been performed in vitro, but there are not many good models, in vitro models allow us to recapitulate spermatogenesis. Another thing is that CRISPR screen is really good at finding cell intrinsic effects, meaning that when you perturb a gene within a cell, you will know what happened to the cells that got perturbed, but it wouldn't tell you what happened to the cells spatially closer to the cell that got perturbed, because you are using single cell data. There's no spatial context with single cell approaches. So that means there are significant challenges associated with applying CRISPR screen to understanding spermatogenesis. So what we are proposing is pretty simple. What if we could perform CRISPR screens in vivo in the native context of testes, and in the meantime, read out those perturbations using spatial transcriptomics? It seems to be a very simple concept and no-brainer, but in reality, when we try to implement them, there are tons of hurdles. But at least now, we're happy to report that we were able to establish a pipeline that allows us to really introduce a pool of guide RNAs into testes in vivo using testicular injections of adeno-associated viruses. And then we are working to use a spatial transcriptomics approach to capture those guide RNAs along with their the transcriptome of individual cells using the approaches I previously described to you. Hopefully next year I will be able to show the full results of this project. And with that, I'd like to thank my whole lab who has done all the great work that you've seen today, and also our collaborators, especially Makoto and Kyle, who have been super helpful in terms of SSA transplantation. I also like to thank my funding source, especially NICHD, to support us to develop those in vivo CRISPR screen approach to understand testicular gene functions. Lastly, our lab is growing. We are looking for trainees at all levels, graduate students, research technicians, and postdocs. So if you're interested in our work here, please feel free to reach out. Thank you so much for your attention. Happy to take any questions from there.
Thank you so much for giving us such a wonderful talk, Haiqi. And all attendees are now very welcome to type your question in the Q&A box. We already have one question there. What type of analysis did you perform to identify the ligand receptor interactions on the public single-cell RNA sequencing?
Right. So there's a data package called niches that we mainly took advantage of that framework. We did some customization. I'm happy to talk about it offline if you're interested. But basically what it did was to calculate the strength of ligand receptor interactions by multiplying the gene expression, normalized gene expression of a ligand and the normalized gene expression of a receptor. And then so that's basically it gives you the so-called strength of the ligand receptor. And then you can do that for each cell pairs. And of course, you have to first start by building a spatial network, meaning that okay, for this single cell, you're looking at maybe 4 nearest neighbor of the cells that you're interested in, and then you sum all the single ligand receptor interactions among different cell types to allow your cell type level integration. But anyways, those are minor things. And again, I'm happy to share the details offline, and the code that we use to calculate those are publicly available. I'm happy to share that as well.
Sure, thank you, Haiqi. The next question is a great talk. How about low expression genes detecting using the Curial spatial technology? Are you able to see low expressing genes?
Well, that's another good question. Well, I think this is this is caveat for all the spatial, maybe not even spatial, even single cell technology as well, that you always have drop-offs of lowly expressed genes. Right now, the capture efficiency of spatial data, especially in the case of a slice-seq, is only about a half or even less of that of the single cell, meaning that you're not capturing a lot of unique transcripts per cell. That being said, one way to overcome this is that you may want to explore alternative approach if you have specific genes in mind. There are targeted approaches where you could use in situ sequencing or other approaches to other similar spatial approaches, image-based spatial approaches to really boost the sensitivity of your data. So that might the overcoming some sparsity issue that are associated with those array-based approaches.
Great. Excuse me. Our next question is from Faye, actually. Did you see enrichment of the lentivirus around the retitastis? How could you evenly deliver the virus in majority part of the testis?
Right. So yeah, that's an interesting question. Well, I mean, right now we're using testicular injections through the retitastis. So we haven't looked at the retitastis per se, but I'm sure it will be there. In terms of the even distribution of the viruses, right? That's another thing that we cannot 100% to solve, just because it really depends. It really depends on when you push the needle, how the viruses can go through the seminiferous tubules. One way to overcome it is that we are not profiling just one plane of the testis. In this case, we are really looking at the whole testis. For example, you are sampling different planes of the whole testis, and also you are sampling different mice from different injections. So that would kind of compensate for the biases that might be introduced by injection itself. Hope that makes sense.
Sure. Thanks, Haiqi. So I have a question. Are you also going to able to identify the interactions of the ligand receptor interaction, like even including the autocrine, paracrine, or even endocrine signaling, because the signal transduction pathway is so complex, including upstream, central mediator, and also the downstream pathway?
Right. That's another good question, Lee. I have to say that there are definitely caveats associated with the ligand receptor interaction analysis that we're currently presenting here. One thing is that we made a fundamental assumption that the ligand receptors happens within very limited spatial distance. So meaning that you really cannot look for long distance interactions. Those, for example, like you mentioned, those upstream hormonal interactions. Maybe that's not the thing that the spatial can do. One thing could potentially overcome that is to curate a list of those long scale interactions and try to put that in mind when we do the analysis. Another thing that we think we potentially do, not for downstream, but for not for upstream, but for downstream, is to identify the downstream signaling cassettes, especially at the transcription factor levels. What happens when you have this enrichment of ligand receptor interactions within the cell? So that potentially can be done, and I think there are already computational approaches that are trying to solve that issue, because it has been demonstrated in single cell. So you would imagine that it would be feasible in the spatial data as well.
Okay, thank you, Haiqi. For such a wonderful talk. Yeah, for all attendees, if you have additional questions, you are really welcome to type your question, because Haiqi will stay for 30 minutes longer, and you can communicate. You can even like email Haiqi for other questions and talk it offline. So now thank you. Yeah, now let's welcome our second speaker today, Dr. Fei Zhao, assistant professor from the University of Wisconsin in medicine. So Dr. Zhao is an assistant professor in the Department of Comparative Bioscience at the University of Wisconsin Medicine, and he received his PhD from the University of Georgia under the guidance of Dr. Xiaoqin Ye. Actually, we graduated from the same PhD lab. And then he completed his postdoc training in Dr. Humphrey Yao's lab from the National Institute of Environmental Health Science, or NIEHS. And Dr. Zhao's current research is primarily focused on elucidating the cellular and the molecular mechanism underlying the sexual differentiation of reproductive tract development. And today, Fei will introduce his research. He opens his lab in the past 2 or 3 years. And Fei, I will turn it over to you.
Okay. Is everyone can hear me?
Yeah.
Okay, perfect.
And your slide looks great too.
Okay, let me see. Pointer. All right. Hello, everyone. Sure, thanks for your kind introduction. First, I would like to thank the SSR committee for such a wonderful opportunity to share my research with you today. I really enjoyed Dr. Hai-Chun Qi's talk in the first half of the hour. Now, I'm going to spend some time introducing my research program at the University of Wisconsin that aims to understand sexual differentiation of reproductive tracts. I know in this cohort, and we are all interested in the study of reproduction. However, we work on different aspects using all different kind of models. So I would like to first familiarize some of you with the mouse reproductive tract. And we are using mouse as an experiment model in our lab. So the mouse, in the male, you have the testes, so where the sperm produced, and the male reproductive tract, which include epididymis, vas deferens, and the seminal-vesicle. So the male reproductive tract are critical for sperm storage, maturation, and the transport. By contrast, in the female, you have the ovary, and female reproductive tract, which include oviduct, uterus, cervix, and vagina. So the female reproductive tract provide the site for fertilization and play critical role for embryo transport, development, embryo implantation, and placentation. It also provides a passage for delivery during parturition. So the male and the female have morphologically and functionally distinct reproductive tract. However, early on, before sexual differentiation, regardless of male and female, the embryo have the same setting. So both male and female, and before sexual differentiation, they have a pair of undifferentiated gonads, and then two sets of paired reproductive tract. The Wolfram duct, the progenitor for male reproductive tract, and the Maroon duct, the progenitor for female reproductive tract. So during sexual differentiation, the gonad in the male differentiate into testes, which produce two hormones, androgens, that promote the survival and the differentiation of Wolfram duct, and the other hormone, anti-Muller hormone, MH, for inducing the regression of the Muller duct. So everyone, what's the consequence of this? What's the consequence of the action of the two testicle hormone actions? So as a result of the action of two testicle hormones, you would see the Wolfram duct is maintained, while the Muller duct is regresses in male. So the maintained Wolfram duct will then give rise to the male reproductive tract I just introduced in the last slide. By contrast, in the female, the gonad develops into the ovary, which does not produce these two hormones. Consequently, the Wolfram duct degenerates in the absence of androgen action, and the Muller duct is maintained in the absence of anti-Muller hormone action. And then the maintained Muller duct are going to give rise to the female reproductive tract I introduced in the last slide. So the sexual differentiation of reproductive tract really depends on the testicular hormone actions. With the hormone actions, you are driving the male phenotype. And the female phenotype arises in the absence of those two hormone actions. Therefore, is considered a default or passive pathway. If you make a cross-section of those tissue, okay, for example, if I make cross-section of this fetal tissue known as mesenterophores, you will notice that these two ducts actually surrounded by their own mesenchymal tissue, which is undifferentiated connecting tissue. So Wolfram duct mesenchyme and the Muller duct mesenchyme. Those two mesenchyme are different, expressing different markers, and they have different properties. So it's well known that mesenchymal syndrome control the fate and differentiation of the reproductive tract. For example, androgen works on androgen receptor expressed in the mesenchyme in are inducing Wolfram duct maintenance. Anti-malone hormone MH acts on anti-malone hormone receptor type 2 for inducing malone duct regression. So in other parts of in other organ development, it's also known that mesenchyme and regulate the fate and differentiation of ductal epithelium. So we have been looking for novel mesenchyme factors. And when we postdoc in Humphrey Yao's lab, we identify COOP-TF2 is specifically expressed in the mesenchyme surrounding wolfin and malone duct in both male, female and male embryo. So COOP-TF2 is an often nuclear receptor, and it have been shown play critical role in the development of other organs. So the specific expression of COOP-TF2 in the mesenchyme suggests its potential role in reproductive tract differentiation. So to address this question, we really want to knock out COOP-TF2 to see what happens. However, COOP-TF2 global knockout leads to environmental lethality. So to overcome that problem, so we generate and design a tissue-specific condition knockout. So we confer, we delete in this condition knockout, we delete COOP-TF2 in the mesenchyme before settle differentiation successfully. Then we collect those tissue to examine the consequence of COOP-TF2 ablation in the female. What you look at now is hormonal immunostaining of PAX2 is epithelial marker, which help us to visualize the malone duct. So here is the ovary, here is malone duct. So there's one single reproductive tract malone duct in the control tissue. In the absence of COOP-TF2, however, and there's two reproductive tracts, both wolfin and malone duct are maintained. You may have a question, how do I know the SSR duct is actually wolfin duct? It could be duplication of malone duct. Well, we confer identity of this duct is indeed ectopically maintained wolfin duct by staining the wolfin duct specific marker. As I mentioned, wolfin duct maintenance is exclusively linked with androgen action. Then you may wonder what happened to sterogenesis in the gonad. And we examine the transcriptome of the control and knockout ovary. We do not see any differential expressed genes. And we'll also examine some sterogenesis enzyme expression. There's no ectopic androgen. There's no ectopic expression of enzyme for androgen production. So you will notice that the ovary shape actually changed in the knockout. This is probably due to the physical presence of the SSR duct. And however, the key thing is the ovary here, the knockout, do not produce any androgens. We also examine androgen response gene in the tract, and there's no androgen response gene elevated in the knockout tissue. So those results suggested that the wolfin duct maintenance in knockout female is not due to androgen actions. And then we wonder what could be the mechanism. So how does the absence of COOP-TF2 in the mesenchyme lead to wolfin duct maintenance in a female embryo? So we turn our attention to local growth factors. And among several growth factors we investigated, we found in the absence of COOP-TF2, FGF expression is increased. So we perform qPCR to examine the FGF receptor and the major ligand expression, identify FGF 7 and 10 are significant increase in the knockout. So FGF2 is known can act on its receptor, expressed in the epithelial cell to turn on intracellular pathway mediated by pERK and pAKT. And among those whole pathway, we identify the pERK actually is activated. So we examine pERK expression that in the epithelial cell in knockout. So the specifics, so there's enhanced pERK expression. Then we ask, what's the functional significance of the pERK mediated pathway? To address that question, we actually collect the knockout tissue and then culture them in the ex vivo condition. We culture in the presence of vehicle or in the presence PERK inhibitor. We argue if PERK-mediated pathway is very critical, and then the PERK inhibitor could be partially or completely rescue the phenotype in the knockout female. So in the control group, in the vehicle treatment group, the knockout tissue have the Wolfram domain 10, just like the in vivo situation. In the knockout, in the presence of the PERK inhibitor, okay, the Wolfram target is eliminated. Only one single murder target is maintained. So rescuing the phenotype of the knockout, CoopTF2 knockout. So those results suggested that the CoopTF2, in the absence of CoopTF2, there is enhanced FGF production that leads to PERK mediated survival pathway in the Wolfram target epithelium. So those is observation in knockout tissue. What these results tell us, what the function of CoopTF2 in normal condition. So in normal condition, the CoopTF action is to suppress FGF production to safeguard Wolfram duct regression in the female embryo. This is in contrast with the current notion that Wolfram duct regression is a default and passive outcome. It occurs due to absence of androgen action. Instead, our results demonstrated that the elimination of Wolfram duct in the female embryo is actually actively promoted by the CoopTF2 in the mesenchyme. Everyone, if you're still with me, you may have the question. So the Wolfram duct survival is promoted by androgen signaling, androgen receptor signaling. Now I'm telling you, CoopTF2 actually suppress Wolfram duct survival. You may wonder how the AR and the CoopTF2 interplay in controlling the Wolfram duct development. So this is a question we have been doing, and we have some clue, and we perform CoopTF2 and the AR AR chip-seq to identify their genomic binding in the reproductive tract. To our surprise, their genomic binding do not overlap. So they do not overlap, and their downstream gene do not overlap that much. So those results suggested androgen receptor and CoopTF2 may induce two different or separate pathways to control the fate of the Wolfram duct. Okay, after the discovery of the critical role of mesenchymal CoopTF2 in the Wolfram duct mesenchyme, we remain our interest in Wolfram duct mesenchyme, but ask a different question. I hope you're still here and still know that in a female, okay, before sexual differentiation, there are two ducts, the Wolfram duct and the Mullern duct. They're surrounded by their own mesenchyme. So Wolfram duct mesenchyme, Mullern duct mesenchyme, which actually express the marker GLI1 and MHR2 respectively. I told you during Wolfram duct degeneration, it's controlled by the Wolfram CoopTF2 in the mesenchyme. Okay, in the female, the Wolfram duct degenerate. And then we ask, what about the surrounding what about the surrounding the mesenchyme? After Wolfram duct epithelial cell degenerate, where is the Wolfram duct go? So what's the fate of the Wolfram duct mesenchyme in the female reproductive tract? Are those mesenchyme eliminated along with the epithelial cell or become part of the female reproductive tract? So given the importance of mesenchymal cell in organ development, we actually designed a linear tracing model, tamoxifen inducible GLI1 Cre tomato, that we can label and trace the fate of the Wolfram duct mesenchyme as they differentiate. So in this model, we inject tamoxifen before sexual differentiation, label some of the mesenchymal cell, and then trace where they are during differentiation. So after 2 days, and Wolfram duct degenerate, and at the time of birth, at the embryonic day 18.5, and Wolfram duct degenerate, completely degenerate. However, those labeled wolf and mesenchyme actually maintain and still become part, maintain and become part of the female reproductive tract. And one thing I want to very important to point out, actually, all my image is arranged in a way. The upper part is mesometrial site, so the lower part is anti-mesometrial site. So if you look at me, my camera. So the uterus, for example, the female tract is a tube. So only one side actually connected to the body cavity and systemic circulation. That is known as mesometrial site. And the other side is known as anti-mesometrial site. So why this matter? Well, there's some the physiological events in uterus, for example, have some location bias. For example, implantation always occur in mice, always occur at the anti-mesometrial site, while presentation only occur at the mesometrial site. So the position is very important role. So if you could notice that, if you look very carefully, you will notice that the wolf-and mesenchyme derived cells are predominantly localized at the mesometrial site. By contrast, if you label using MHR2 Crayosa tomato to label Murinda mesenchyme, those mesenchyme derived cell is more surrounding the Murin duct. As they differentiated, they are localized at anti-mesometrial site and bilateral, but seldomly found in mesometrial site. So those results demonstrate that wolf-and mesenchyme contribute to an anatopically unique mesenchyme population in the female reproductive tract. And then we ask how those two mesenchyme populations different. So there's two mesenchymal cells form with the mesenchymal compartment in the female reproductive tract. So how those two mesenchymal cells different? We actually use the linear tracing model that I introduced last slide, and then enrich those two populations and perform RNA sequencing. I'm not going to show you the over 1,000 differential expressed gene. Instead, I'm going to show you the two marker we identify for those population. So first, we identify the AKAP12 is a kinase occurring protein. It's a specifically expressed at the mesometrial site. That is where wolf-and mesenchyme derived cell localized. By contrast, the HAN2 is a transcription factor. It's specifically expressed bilateral and anti-mesometrial site, but not in the mesometrial site. So this result demonstrated that wolf-and mesenchyme contribute to not only anatopically, but transcriptionally unique mesenchyme population. And then we ask, what's the functional significance of wolf-and mesenchyme in the female reproductive tract development? To address this question, we are using GLI1 Cray-ER-ROSA DTA, a genetic cellular ablation model. So in this model, DTA is a toxin. When you give it tamoxifen, you actually turn on DTA expression, specifically in the GLI1 positive cell at the time of tamoxifen. So we can induce the expression DTA and cause the apoptosis of those cells. One thing you may worry, after the apoptosis, the toxin may release to the surrounding environment. Well, the DTA, that's not the issue, because DTA, endocytosis of DTA will require a receptor which is not expressed in mouse cells. So we can actually specifically target and delete GLI1 positive wolf-and mesenchyme at the time of tamoxifen treatment. And then there's another issue: GLI1 expressed everywhere in the body. If you perform in vivo, that will lead to embryonic lethality. So we get the tissue out and culture them perform ex vivo organ culture. So this is control tissue, this ablation group. So at the beginning, there's no DTA treatment, there's no DTA expression. Upon given tamoxifen treatment and DTA turn on in the control group, there's no ablation. You see the coiling of the oviduct and expansion of uterus. However, in the ablation group, the coiling is decreased in the oviduct region, and the lumen expansion uterus is also diminished. So this result demonstrated that wolf induct mesenchyme is very critical for female reproductive tract development. So far we have talked about the wolf induct mesenchyme, its critical role in promote wolf induct degeneration and contribute to part of the female reproductive tract. So it can be designated as the female fate of the wolf induct mesenchyme. It is known that if you provide exogenous androgens in the female, you actually can masculinize the wolf induct and promote wolf induct metabolization, maintenance and differentiation, just like its counterpart in the male embryo. So in the male embryo, it's known as, it can be designated as a male fate. So our understanding of wolf induct mesenchyme is not complete until we understand both male fate and its female fate and male fate. So that in the male, we know androgen play a super critical role. They drive the formation and differentiate. They drive the maintenance and differentiate of wolf induct. And the influence of androgens, wolf induct first stabilized, and then undergo coiling at the cranial part, and then extensive coiling in the body and the caudal part. So I mentioned earlier, people, it's known that the androgen receptor in the mesenchyme is critical for the androgen action in driving wolf induct development. However, we still don't have the direct genetic evidence. So to provide the genetic evidence that androgen receptor in the mesenchyme is critical, we need mesenchyme specific array. So we spent quite a lot of time to search and test and array, and we finally got one. It's actually used in craniofacial development. However, it's also specifically expressed in is still very active in the mesenchyme surrounding the wolf induct. So when we use this array to cross the AR, we are able to specifically delete the androgen receptor in the mesenchyme. So here is the immunohistochemistry of androgen receptor. So at control group, AR is expressed in the mesenchyme at the initiation of sexual differentiation. This is consistent with the literature. So in the knockout, in the conditional knockout, and you see an androgen receptor is totally abolished, eliminated in the mesenchyme. As interestingly, androgen receptor start expressing in the epithelial cell. We still don't know why, and that's something we're still working on. But the key message is wolf induct mesenchyme is successfully deleted in this model. So in the control group, and the androgen and the influence on AR in the mesenchyme, the wolf induct will undergo morphogenesis, become very coiled. By contrast, in the absence of androgen receptor in the mesenchyme, the wolf induct partially degenerate. The remaining wolf induct actually become cystic. So we are excited. This is actually first direct genetic evidence that androgen receptor in the mesenchyme is indeed critical for wolf induct development. This is actually also consistent with the notion that androgen is also the major driver for masculinization. What they're looking at now is a landmark or critical study in understanding androgen-driven masculinization. So from discovery of testicular secretion and the effect of those secretion, and the introduction of hormone concept, and isolation purification of testosterone, and cloning and crystal structure of androgen receptor. Now, in recent years, we're using tissue-specific knockout mouse model to understand androgen signaling in specific cell. So the last slide is actually belong to the tissue-specific function study. So everyone, which year are we in now? 2024, which you can divide it by 4. So it is a leap year. I like leap year because in this year, we are going to take a leap in understanding wolf induct development. We are trying to identify any new players in wolf induct differentiation. There is definitely multiple ways to do that. So here I have, I just introduced one way we're doing now. So we actually try to identify new marker by assaying chromatin accessibility landscape. So the DNA wrap around the histone. So in order for the androgen receptor to bind the regulatory region, they had to become accessible. So we argue we can actually to survey the chromatin accessible chromatin region to deduce any potential new transcription factor during masculinization. So the experiment is quite simple. We actually get the tissue and enriched wolfendam mesenchyme from the male and the female, where there's androgen and where it's no androgen action, and then collect tissue at the 2 time point at the 14.5, 16.5, this window of sexual differentiation. And then we identify over 1,000 of accessible chromatin region and then deduce the enriched motif in this region. Due to the time constraint, I just want to show you that we work on the GAT2 now, GAT family. Among those 6 members in the family, we identify GAT2 specifics expressing the mesenchyme. So when we delete the GAP2, so in the mesenchyme, okay, so in the control, we have very coiled epididymis, but in a knockout, the coiling only occurs at the cranial part and becomes straight tube in the body and the caudal part. And we try to understand the interplay of GAT2 and the AR. At the same time, we know paracrine growth factors is mediated the mesenchymal action determine the fate and differentiation of the epithelial cell. So we identify the well-known established morphogen or paracrine factors that play critical role in epididymal coiling, inhibit beta A. In normal condition, expressed this robust in the mesenchyme surrounding the Wolfram duct. But in absent GAT2, its expression seems to be slightly reduced. The more critical thing is this expression pattern changed. Instead of in a mesenchyme adjacent to Wolfram duct, is now expressed in the outer layer of the mesenchymal cell. So we are still to find out what happened. So what's the implication of those change in this phenotype? Okay, with that, I want to summarize what we discussed today. We're working on Wolfram duct. First, we demonstrate the Wolfram duct degeneration is not default. It's controlled by COOP-TF2 in the mesenchyme, and they contribute to part of female reproductive tract. And in a male, we provide evidence that the androgen receptor is critical and identify new factor GAT2 that play a role in Wolfram duct masculinization. So with that, I would like to thank my lab people and former lab people and the grant support. Thank you.
Thank you for another wonderful talk, Fei. And I think that I need to steal some of your data to update my slides for teaching the sex determination in my physiology class for the pharmacy student. So yeah, all attendees, you are very welcome to type your question in the Q&A box. And yeah, I can kick off. So when you say the ovary, it seems it's ovary dependent, but you just mentioned it's based on your like a single cell sequencing data. But did you measure the hormones or have more like evidence beyond the transcriptional level?
Yeah, so the ovary, we actually we haven't measured the androgen level, but we do measure the androgen dependent endpoint. For example, anal genital distance. So in a knockout female, anal genital distance is comparable with the normal female. So that's a good indication there's no androgen ectopic androgen production in a female.
I see. So as a reproductive toxicology, I always wonder whether like during pregnancy, the exposure to endocrine disruptors, no matter androgen or estrogen lag, or they can target on their nuclear receptors, whether will play a role. So what is your vision regarding that? Whether they will totally turn it on or off, or make it messed up, or like a kind of in the middle between the male and the female fate?
So I think that will be your expertise. But my sense, my piece of sense is that it's really depending on dose and timing. So the time whether you treat the same treatment will, for example, masculinization had to occur at a specific window, even though you provide androgen, very high androgens, but if you do not treat at the right time, they probably no effect at all. So dose and timing is definitely critical in determining the toxicity.
So there's a threshold. Yeah.
Yes.
Yeah, we have a question, a great talk. If you force COP-TF2 expression in males, will the Wolfram duct regress? And what about in the presence of androgen signaling?
Yeah, so in the male, COOP-TF2 actually expressed in the male. So basically, in the male COOP-TF2 they should be suppressed Wolf duct. However, the androgen signaling override COOP-TF action. So androgen receptor signaling is still dominant and promote Wolf duct survival and differentiation.
Got you. Yeah, thank you, Fei. Yeah, all attendees, you feel free to email Fei and talk offline. And at last but not least, I would like to thank both speakers for the great talks today. And we also want to thank all of the attendees for joining us today too. And thank you so much. You have a great day.
Thank you.
Thank you.
Bye.